Welcome back to our channel. In the last video, we demonstrated how to perform homology modeling through the automated mode. In today's video, we shall discuss how to perform homology modeling via the manual mode. Manual homology modeling is required to overcome the limitations of automated tools, particularly for complex regions like insertions, deletions, and loops, and to improve the accuracy of the model by refining sequence alignments and optimizing the placement of sidechains. Manual input is crucial when automated methods produce potentially erroneous models due to the need for an expert's judgment in handling challenging alignments or when high-resolution predictions are needed for detailed applications such as drug discovery or understanding protein-ligand interactions. Okay, let's delve into the manual mode of homology modeling. Using your internet browser, either Google Chrome, Firefox, Microsoft Edge, Internet Explorer or Safari, click on the link displayed on the screen. The link is also posted in the comment section for easy access. That will take you to the website displayed here. Paste the first protein sequence. Let's use the p53 protein that we have already used in our previous videos. You can either paste the FASTA sequence directly or upload it from your computer, as indicated. Under Database, select the database as Protein Databank, PDB. Click on Blast. Once you get the results, scroll down. Click on the Per Ident heading of the table. You will see something like this. Click on First, Accession No, of the result table. That will take you to this page. Click on the FASTA. Copy the FASTA sequence and paste in Notepad. Save it as template protein1.txt. This is our template sequence. Now, we want to do the alignment. We shall use the Clustal Omega software. Click on the link displayed on the screen. The link is also supplied in the comment section for easy access. That will take you to the site displayed on the screen. Scroll down and click on the Clustal Omega under the Multiple Sequence Alignment section. Paste your protein sequence with its FASTA heading on the assigned box. Paste your template sequence with its FASTA heading directly below the protein sequence. Click on Submit without changing any parameter. After getting the results, click on Download. You will get a file like the one displayed on the screen. Remove the Clustal O 1.2.4 multiple sequence alignment line on top. Save the rest of the file on Notepad as target protein. ALN. Save file type as all files. Now, we will do model generation. For this, click on the link displayed on the screen. The link is also supplied in the comment section. After clicking the link, you will come to the Swiss model website. If you are given options, select the first biounit. Click on Build Model. You will get a result like the one displayed on the screen. Click on the first build model. Once you get the result, click on the download icon. A zip file will be downloaded. Extract the zip file. You will need the model, PDB for model validation. Now, you can click on the different icons to get more information concerning the outcome of your job. For instance, you can click on Project Summary and get a summary of your results as presented on the screen. The results shows that the target protein and the template matched each other. You can try different proteins to see their homology. The colored parts indicate where the amino acid residues of the target protein and the template match each other. Where they do not match are shown in dashes. You can click on Project Data and choose Project Report Display. This will display the report of your project on the screen. You can click on other icons like the one here to see the similarity between your target protein and the template. The results would also be sent to your supplied email address, and you can click on the link provided to see the results. In today's video, we discussed the manual mode of performing homology modeling using the BLAST, FASTA, Clustal Omega, and Swiss model tools. You can use this homology modeling exercise to compare different protein sequences and know their similarities. Likewise, you can compare DNA sequences through the homology modeling techniques. And that is it for today. In our future videos, we shall demonstrate how to induce mutagenesis, point mutations. There, you will learn how to perform in silico point mutations and predict if the results would be silent, missense or nonsense mutations, based on the site of mutagenesis. We shall also discuss how to construct phylogenetic trees for both DNA and protein sequences. We shall likewise demonstrate how to generate RNA primers for DNA synthesis and learn the various molecular docking techniques for protein-protein and protein-ligand interactions. Therefore, watch out for our new videos. If you have found this video useful, kindly like, comment, share, and subscribe to the channel so that you can get notified whenever new videos are uploaded. Bye for now and see you in the next video.